So today we're going to look at the process of mitosis. So if you guys remember, mitosis is a form of asexual cell division. It occurs in every one of your body cells. Basically, we're looking at one cell is going to produce two identical daughter cells. So that parent cell is our original cell. These daughter cells are going to be the new cells that we're forming through this process of mitosis. And we said that we're going to start out with what is known as a diploid cell, and we're going to end up with two cells that are diploid. And what diploid means is that it has a full set of chromosomes. So if we were doing this with a human cell, let's say a skin cell, that diploid number or 2N number would be 46, because you would have 46 chromosomes in every one of those skin cells. Here we're going to deal with two chromosomes. So I have a red chromosome and I have a yellow chromosome. These parts are going to be important for you guys to know, because as we get into meiosis, you guys are going to kind of see this taped again, those who are home, and you guys are going to kind of voice over my part for me and send that back into me. Those of you guys in class are going to be actually physically working with beads and presenting to me one-on-one within class time. So the beads that you see are representing our chromosomes. These individual beads that we have here are considered our genes. So we can say, well, these should be completely identical in terms of chromosome, because it would be chromosome pair number 1. In the middle, we have our centromeres. So centromeres are joining together the parts of our chromosome. Out here in the cytoplasm that we're going to consider my tabletop, I have centrioles. I have other organelles. So I have mitochondria and Golgi bodies and endoplasmic reticulum and ribosomes, and I have all these other cell parts out here. What am I worried about in this particular cell? We're considering this is going to be my animal cell. We're worried about our centrioles. So if you remember centrioles, they are important in cell division. It's what my spindle fibers are going to attach to. They are only found within your animal cells. So we're not going to see these within plant cells. Plant cells have a really rigid cell wall that these spindle fibers can kind of attach to for us. In the middle here, I have this baggie, and this bag that you see is going to represent my nucleus. So inside my nucleus, I have my genetic material, in this case, 2 chromosomes, whatever made-up organism you want this to be. This organism has 2 chromosomes in every one of its body cells. So as we look at the process of mitosis, we first want to start out with the beginning part of it, of the cell cycle, which is going to be interphase. So a lot happens in interphase that we need to know about. And the good part is that interphase is going to be the same whether we're talking mitosis or we're talking meiosis. So as we think about these particular genetic material inside my nucleus right now, normally in a non-dividing cell, you're not going to be able to see your chromosomes. They're not condensed, they're not thick, they're not visible at that point. When you look underneath the microscope and you can see chromosomes, maybe you did it in first year bio and you saw onion root tip and you saw all these chromosomes and they were condensed and they were easy to see, that means that that cell was physically going through mitosis, cell division, at that particular point. Normally, let's say skin cells, they only do this once every about 28 days. So those other 27 days that they're just kind of hanging there, these chromosomes are not visible. So they are long, they're thin, and they're invisible, and they're called chromatin. So chromatin is basically your relaxed form of your chromosomes. That cell goes, oh, you know what? I need to go through and make more copies of myself. And when that happens, that's when we're going to start to see the chromosomes. So right now we are in the beginning of our cell cycle, and the cell cycle enters into interphase. Interphase is not a true phase of mitosis. It is kind of this getting ready phase. People used to think kind of was this resting phase because nothing really happened. Well, there's a lot of stuff that's going to happen here. So this is actually the longest part of the cell cycle. It can take up to about 18 hours of maybe our 22 hours of the entire process. What needs to happen is the cell is growing, it's getting larger. Remember, this cell can only control so much cytoplasm. So as a point it gets too large, it's going to stimulate that whole process of cell division. My organelles need to start replicating during this particular time. I'm going to be replicating my genetic material, so a lot going on. Interphase is divided into 3 parts, G1, S, and G2. So G1 is kind of this first gap phase, this kind of growth phase. The cell is getting larger. So the chromosomes are going to start to become a little bit more visible at this particular point. And what we're going to see is that my centrioles are going to go through and they're going to replicate. So my paper clips, I have 2 paper clips because centrioles always come in pairs. There's my 1 pair. They're going to go through and make a copy or replicate themselves. Mitochondria are, and Golgi bodies, and all my other organelles are. Remember, this 1 cell is now going to become 2, so we need to make more organelles. So my centrioles, which are represented by my paper clips here, have gone through and replicated. They are not in the nucleus. They're out in the cytoplasm hanging out. The second phase of interphase is called the S phase, which is synthesis, which should be familiar to you guys. I'm making more. Well, what am I making more of? You did this in DNA and protein synthesis. We replicated DNA. DNA gets read, 3' to 5' gets made, 5 to 3. We're going to do an easy way. I'm going to make identical copies. These little centromeres in the middle are kind of magnets, so they're going to stick together. I have just copied my DNA. My yellow chromosome, I'm going to copy my DNA. So I have made identical copies, but right now I have twice the amount of genetic material inside that nucleus that what I should have. So that's why we're going to go through and divide this cell here in a moment. Then I'm going to go into G2. So we did G1, growing, replicating some organelles. My S phase, I've replicated my genetic material. G2 is that second gap phase. So my centrioles are done replicating. I'm going to start to increase some protein synthesis, and I'm going to start to create something called spindle fibers. So in this little diagram, my fingers are going to be the spindle fibers because they're out here in the cytoplasm. They're eventually going to hook up to my centrioles, and they're going to start to pull these chromosomes apart eventually. So it's these microtubules that are going to kind of connect, and it's kind of like fishing line, I say, because it's kind of like you're going to be fishing and you're going to be pulling apart these sister chromatids in a moment. So we are done interphase. Interphase is a very long phase. So what we need to do is we are now going to go into prophase. So if you remember the steps of mitosis, PMAT, prophase, metaphase, anaphase, and telophase. So in the beginning of prophase, what we're going to see, in the beginning of prophase, first off, my chromosomes become visible for the first time. So they have condensed, they become short, they become fat, and now they are visible. And they are known as sister chromatids. So we started out as chromatin with an N, long, thin, invisible. It went through and replicated while it was kind of invisible there. It then condensed, became short and fat and visible, and now we've got sister chromatids. So each of these is a chromatid. This is a chromatid, this is its replicated piece. So just moving out of the way for a moment, and we're looking just at that particular red strand there, and that red strand, chromatid, chromatid, they are identical pieces. They are known as sister chromatids. So I have a red sister chromatid, I have a yellow sister chromatid. This is what we're seeing in prophase. My sister chromatids, my genetic material is visible now. My centrioles are going to migrate. They're going to move to opposite poles. So if you think of, let's say like a globe, the poles, North Pole, South Pole. So they're going North Pole, South Pole, they're going to opposite ends. Real important, we got to get this genetic material out of here. So what's going to start to happen is that my nuclear membrane is going to start to break down. So I'm just going to kind of slide the baggie away. Nuclear membrane breaks down. My genetic material is now free because we got to separate these and go to opposite poles. My spindle fibers are going to start to attach to my centromeres, and that is kind of the joining point of these sister chromatids. So it's like these long threads, and they're going to hook up to the centrioles, which are opposite poles, and they're going to hook up to the centromere of my sister chromatids. And that's why I said it's kind of like fishing. Think of these centrioles as little fishermen, and they're throwing out their lines, the spindle fibers, and they're attaching onto the centromeres. Eventually, they're going to start reeling in their catches, but not yet. Okay, so right now we're still in prophase. Let's recap. Centrioles moved to opposite poles. Nuclear membrane disappeared. Spindle fibers are starting to form, and they're going to hook to the centromeres. Now, metaphase, middle. What's going to go to the middle? My sister chromatids are going to go to the middle. And who's moving them? My spindle fibers. Let's go this way so I'm not blocking you out with my shoulder here. And they're going to start moving to the middle. So it's kind of like I spindle fibers attached this way to my centromeres and centrioles, and I've got them attached this way. My sister chromatids are lined up at the middle. We don't want them lining up like this. because that means they would have to separate that way. They are lining up like this. The spindle fibers are attached to the centromeres and centrioles. They are lined up along the middle. Now we're going to go to anaphase. Anaphase is away. This is the quickest phase, about 5 minutes total. My spindle fibers attached to my centrioles. Centrioles, my little fishermen, are going to start reeling in their lines. And when they do that, my centromeres are going to split, and my sister chromatids are going to split. As soon as they do, I now have chromosomes. They're going to opposite poles. So my spindle fibers are pulling them in. This is all anaphase. It's going away from the middle towards the poles. Spindle fibers are pulling them in. So anaphase basically ends when they reach the poles. Then we get to telophase. And what we're going to start to see is we're going to start to see these processes known as karyokinesis. We need to start dividing all of our materials on the inside here. So division of our nuclear materials, basically what we're doing there. Now we're going to start to see cytokinesis. Cytokinesis means we're going to start pinching in the cell membrane. So as we work our way into telophase, our chromosomes have reached opposite poles, and basically everything that happened in prophase is going to undo itself in telophase. So these guys are like, hey, you know what? My job is done. I don't have to be all condensed and tightly wound anymore. I can kind of relax and become long and thin and invisible, and I'm going to become chromatin again. So my chromosomes are going to relax. They're going to become long, thin, and invisible. They're going to become chromatin. My nuclear membrane is going to reform. So I have 2 cells that I'm forming. So I have 2 baggies here that are going to form my 2 nuclear membranes. Centrioles, centrioles, nuclear membrane, nuclear membrane. Well, what has to happen is that my cell membrane is going to start to pinch in, and I'm dividing my cytoplasm. So what's going to happen is that this cell is going to completely pinch off. It's called a cleavage furrow in an animal cell. I'm pinching the cell membrane, and what I end up with are 2 cells, separate cells, with a diploid number of chromosomes. So what that means is I started out a cell with 2 chromosomes, 1 red, 1 yellow. Now I have 2 cells, 2 daughter cells, that are diploid with 2 chromosomes in them. 2N is my diploid number. I still have a yellow and I still have a red. Over here I have a yellow and I have a red. These 2 cells are completely identical. They have the same genetic material, unless there was a mutation, which we do not see. Spindle fibers have disappeared at this particular point. My genetic material is chromatin, and these 2 cells are kind of hanging out. If this was a plant cell, some differences that you guys would have seen along the way. First off, we've already mentioned it. You would not have the centrioles. You still have spindle fibers. You have these little asters, which are like little anchors, and these spindle fibers will still pull apart everything else that we saw. The next thing is, I can't form a cleavage furrow. I can't pinch in that cell membrane, or pinch in the cell wall on my plant cell. Animal cell, real soft cell membrane, you pinch it in, it breaks. I have 2 cells completely identical. Your plant cell is rigid. It has cellulose on it. It's like building a brick wall around something. So it's not like that brick wall can be pinched in. So no cleavage furrow forming in a plant cell. What has to happen is that plant's going to take extra cellulose and start to form what is known as a cell plate. So once again, let's go back to that onion root tip you looked in first year bio. It looked like a bunch of bricks that were stacked up that you could see those particular chromosomes on. That is because of that cellulose that's forming those cell plates between these particular cells. But the things that are identical here between plant cell and animal cell mitosis, we have to go through the same steps: interphase, prophase, metaphase, anaphase, and telophase. Those are the steps of the cell cycle. Remember, interphase is not a step of the cell cycle. I started out with 1 parent cell. So this was basically my parent cell. It looked like this. 2 chromosomes in it. By the time I'm done, I end up with 2 cells that are completely identical. Diploid number is 2, so I go from diploid cells to diploid cells. This is what's occurring in all body cells. When we get to meiosis, you're going to see that the only cells that are going to be going through meiosis are gametes, are sex cells. Sperm and egg are created through that particular process. So we're going to go through this a few times. You guys will see a little interactive assignment with mitosis. And the good news is, once you get down these steps, prophase, metaphase, anaphase, telophase of mitosis, you're going to see them again in meiosis. But we're going to kind of go through the process twice. Good news is, interphase stays absolutely the same. The words chromatin, chromosomes, chromatids, I know there's a lot of C words. Get them straight. We'll do them a few times within class, and I'll hold you guys up some examples with these particular ones. Once again, when we get to meiosis, you will see this again with meiosis, except I won't have any sound to it. You guys will kind of be voicing over sound to it and sending it back to me. Okay, so stay on top of this stuff. Any questions, let me know.